Cell culture. Fish culturing you might be doing. Like, what is fish culture? So exactly same thing we do in cell culture also. In the case of fish itself has a certain abilities, isn't it, to overcome the infections. Because our body has a certain measures to overcome infections. So what are they? Now, phagocytosis is there, isn't it? So one type of white blood cells engulf the microorganisms that enter into your body. Then next, the entry will be prevented. That is the primary defense mechanism. The entry of microorganisms into your body will be prevented by skins and by skin as well as mucus layer. Mucus layer will be there in internal tubes which is exposed to external environment like trachea, your intestinal tract, etc., isn't it? And also these internal defense mechanism also involves what else? The antibodies, isn't it? Antibodies produced by what type of cells? What type? B cells produce antibodies, and also T killer cells are there. So all these mechanisms are there in a living organism. But in the case of cell culture, no such mechanisms are there. So how can we overcome that? You can add antibiotics. So normally we add an antibacterial as well as an antifungal agent. So that is the difference from the fish culture for the cell culture. And also we maintain a sterile atmosphere. Even though we are providing or we are adding antibiotics and antifungal agents, if the number of microbes entering into the system is too high, what will happen? These antibiotics will not be able to suppress the microbial growth. So we need to keep a sterile environment. So if you provide all this, you can culture the cells effectively. So this cell culture was started in 1900. First, scientists started culturing tissue. Small pieces of tissue are cultured. So what would be the defects there when you are culturing small piece of tissue? Yeah, the cells which are inside will not get sufficient oxygen as well as nutrients. So that was a problem. And also, when you are taking a small piece of tissue from an organism, whatever microorganisms in that organism is present in the tissue, also, that problem was also there. So basically, the main problem is the internal cells will not get oxygen as well as nutrients, and also the waste materials cannot be removed. So that was a handicap for the tissue culture. So that is why we developed cell culture. So here, you know, in a tissue, the cells are bound together by how? How the cells are bound together in a tissue? Any idea? By protein molecules, isn't it? Extracellular matrix is there, and also certain protein molecules which run across the cell as well as outside the cell. So they help to bind the cell to the nearby cell. As a result, they are held together. So if you want to separate the cells, what do we need to do? Yeah, these proteins which are helping the cells to attach need to be removed. So that is the mechanism. So we use a protease enzyme. Why proteolytic enzymes are used to remove the proteins which help the cells to attach. And for this enzyme to act, the presence of calcium will be a suppressing thing. So we add chelating agents to remove calcium ions from the medium or from the enzyme solution. And you might remember this picture. This is a very famous picture. A wagon tragedy. What happened in there? A number of people were put in a wagon by the British people, and the wagon is fully closed. So there was not enough oxygen, carbon dioxide was not removed, their waste materials were dumped in the wagon, and finally most of them died. So the same thing happens when we culture the cells also. The internal cells will face the same situation. Waste materials, lack of oxygen, lack of nutrients, and they will die. So we are growing the cell outside a living body. And in addition to this nutrients and antibiotic, there should be a surface on which cells can grow. Most of the cells need to be attached for them to grow. And when you are culturing the cell, you can see that. When you are separating the cell from the culture flask using this enzyme, proteolytic enzyme, the cells become round. So if they are not attached, they cannot attain their normal morphology. You know that each cell has a particular type of morphology. Epithelial cell has a morphology, cardiomyocytes has a separate morphology to suit their function. So they need to be attached, except the blood cells. So blood cells, they are suspension. They can grow in a suspended condition. But most of the cells need a medium or a surface for attachment. So for culturing the cells, these are the different steps we need. First, separate the cell from the tissue, and for that we use enzymes. And what enzyme we use? That also varies from the cell to cell. Certain cells are very sensitive, so we use a very mild enzyme. But sometimes most of the cells are quite okay with the trypsin. Then isolate the needed cells. So when you are taking a tissue, another problem is that tissue has different types of cells. Whatever the tissue, there will be more than one type of cell. So we might be needing only one type of cell. So we need to separate that cell. So that is the next step when we are isolating cell from a tissue. Then identify. You have to identify whether you isolate the correct cell or not. Grow in a suitable container. There are different types of containers we use in cell culture. We will see all these containers when you do the cell culture. And provide all the required environment. Required environment means all the nutrients and the oxygen as well as carbon dioxide. Carbon dioxide is also important. Why? If you are providing only oxygen, what will happen? What will happen? The pH of the medium will change. So the cell need a particular pH to grow. Why? What is the need of a specific pH for a cells to grow? Because all the activities in a living body or in a living cell is controlled by enzymes, isn't it? Each and every action needs the presence of an enzyme, and each and every enzyme has a particular optimum pH. You have learned this in first semester in biochemistry. So for that you need to provide carbon dioxide also. Then once you have isolated the cells, what you do? You will grow them in a culture flask, depending upon your need. You can use a small flask if you need only a small number of cells, or if you have more cells, you can use a bigger flask. You have seen different types of flasks here. This is small flask, that is T25. That is the surface area, that is bottom surface area is 25 centimeter square. This is T75. Surface area is 75 centimeter, 3 times of this one. So depending upon the number of cells you need, and also depending upon how many cells you have. If you have only a small number of cells, you will use a small flask, you multiply them, then move to larger flask. And for certain other tests, you cannot use this because this needs a lot of medium as well as reagents. You use multi-well plates. There are also 6-well plate, 12-well plates, 96-well plates, etc. That also depending upon the need of the test. Because if you are using 96-well plate, you need only 100 microliters of the reagent. But if you are using 6-well plate, you need multiple times of the reagent, volume. So you will choose the required type of culture vessel. And the cells are happily growing because you are providing all the required materials for the cells. And what will happen? They will divide. Isn't it? Each cell has a dividing time. Certain cells divide in 24 hours. Some cells divide in 19 hours. Some divide in 34. Some cells don't divide. So, depending upon that, the cells will divide, multiply, and they will fill the bottom of the flask. So after that, what will happen? If you are not removing the cells, excess cells, what will happen? They still continue dividing, and they will grow one above the other. So the same situation comes as in the case of tissue. The lower cells will not get oxygen and nutrients, and the waste materials cannot be removed. So that shouldn't be avoided. So before the cells become 100% covering the bottom, that we call confluency. Fully confluent. Once it is fully confluent, they will grow one above the other. So that needs to be avoided. And also once they grow one above the other, separating them is also difficult, isn't it? Because if you are adding a proteolytic enzyme, that will not reach the bottom of the lower layer. So separating the cell from the flask will be a problem. So better to avoid over-confluence. So before reaching 100% confluent, you need to either remove the cells which are not needed or excess cells to another container if you need more cells, or you can discard it. So transfer cells from one culture vessel to another one. So why? The reason I have said, avoid the cells growing one above the other, isn't it? And if they grow like that, the bottom cells will not get nutrients and oxygen, and the waste materials will not be removed, and your test result will be affected. And how? Because you add, so the cell is fully confluent like this, and you take the flask, you remove the medium. Then you add the enzyme of choice. It can be trypsin, it can be pronase, it can be collagenase, depending upon the cell. And you keep a few, 1 minute or 2 minutes or 3 minutes, depending upon the type of cell, the cell will detach from the flow. And you pipette it a few times, and you will get single cell suspensions. And depending upon that, you will keep a small volume in that and remove the excess. Okay. Then primary cell culture and the secondary cell culture. So, primary cell culture, these are the cells isolated directly from the tissues. Last day we were talking about getting bone marrow. So we are taking the bone marrow of the rat or mice or whatever organism is, and then you are culturing. That is primary culture. Secondary culture or cell line is, you are either getting cells from the primary culture or you are getting from a cell line. Cell lines, they are immortalized cells. They will never die. I think we have learned that cellular aging, isn't it? So after a number of divisions, normal cells will lose their morphology and they will die. But these cell lines, they will never die because they are modified. They are made immortal. So any number of times, we are still using certain cells which were obtained in 1956. So even after 70 or 80 years, still we are using them. So immortalized cells. So secondary cell culture can be immortalized cell lines. And so what would be the difference between them? What care you may need from each type of cell culture? The primary cell culture, what is the possible danger? Primary culture cells you are obtaining directly from an organism. I have already said, whatever microbes are there in the organism will be there in the cells also. So chance of contamination is high. If you are taking cells from a rat, so rat has got hair, isn't it? So the hair is the place where a lot of microorganisms grow. If you are not properly washing the rat after killing with alcohol, all those microbes on the surface will be there in your cell. So that problem is there in case of primary culture. But you don't need to be scared of. If you are very careful, you will not get any contamination or you will get a sterile preparation of primary cells. So cellular senescence, we have discussed this when we were talking about cellular aging. So normal cells, after a few division, lost their ability to proliferate. So they will lose their ability to proliferate and they will not be dividing. Finally, they will lose their morphology and they will die. And because of telomere shortening, you know what is telomere? What is it? Chromosome at the end, there is a random repetition of the same sequences like this, TAGG, again TAGG. So, after each division, the length of this part will decrease. It will become smaller because the end cannot be replicated because of the handicap of the polymerase enzyme. So these are the random repeats, multiple repeats. Sometimes it can be hundreds or thousands of repetitions. So after each division, the length of this part is decreasing, and when it reaches a certain length, the cell division cannot proceed. So that is cellular senescence. So telomere is shortening, that causes senescence of the cells and cells stop dividing. And the telomerase activity can also decline. Because normally there is a possibility that the telomere can be rebuilt. But when this enzyme activity decreases, then it will not be rebuilt and the length will be shortened. Chromosome length decline after every cell division. So cell type oriented culture conditions. Depending upon each and every cell, the need of the cell conditions, culture conditions vary. And why? How? Depending upon the type of cell you are culturing, the culture condition vary. What makes this variation? Depending upon the type of cells, there will be certain changes in the culture medium as well as the culture surface. Certain cells will not attach on the normal culture surface. You may need to provide specific surfaces. Because when we started culturing stem cells, the stem cells were not growing on normal culture plates. So what we did is, we grow a layer of normal cells and then irradiate them. What will happen? Then they will lose their ability to divide. Then we grow the stem cells over those cells. So there was a feeder layer at the beginning of the stem cell culture. But now there are culture plates suitably modified for the attachment of stem cells. We don't need a feeder layer. So depending upon the type of cells, the culture vessel need to be changed and also the medium composition also need to be changed. So cells differ in their basic nature. Epithelial cells are attached. Lymphoblast-like cells are suspended. That is one basic criteria, whether it is attached cell or suspended cells. And also certain cells attach very firmly to the surface. We don't need much modification to the surface of the culture plate. And when they are firmly attached to the surface, what will be the problem? Separating from the flask is difficult, isn't it? Because they are firmly attached. So you need to use a strong enzyme for a longer period of time. But in the case of muscle cells, if we use a strong enzyme for a longer period of time, what will happen? The end plates of the myocytes will be destroyed and their contractile capacity will be lost. So there you may use a collagenase, mild enzyme, or pronase, another mild enzyme. You don't use strips in there. So depending upon the nature of cells, that also vary. Need different added components, as I said before. Certain cells need calcium ions, certain cells don't need any calcium. Like myocytes culture, they are grown in low calcium. Chick embryo cells are grown in higher calcium level. Embryonic bone cells, calcium free medium. So this is one example. So three types of cells, they differ in their calcium necessity. One cell don't need any calcium, like embryonic bone cells, and chick embryo cells need a higher calcium level. Myocytes need a low calcium. This is one example. There are so many other things. Certain cells need glutamine. Certain cells don't need gluten. Certain cells need high glucose content. Some other cells need low glucose content. So if you, for a cells which need low glucose content and you give high glucose, what will happen? It will, whole metabolism will change. So that will give you a raw result. So how do you see the cells? The cells are attached on the bottom of the culture plate. So if you are looking from the top, what will happen? The objective lens will be far from the cells. So instead you look from the bottom. So you use an inverted microscope. And to see the cells in a microscope, what do you do? When you are seeing WBC, different types of WBC, what do you do? You just take blood and put in the microscope and observe staining. Why? Because normal cells are sort of transparent. There is no difference between the different parts of the cells, and you will not see any separate organelles or anything. For that, you will stain. Can we use that staining protocol in cell culture? Certain cases you can use, because if you don't need the cells anymore, you can stain them and see. Because most of the staining process will kill the cells. So that here we use an inverted phase contrast microscope. You should have learned about different types of microscope. Phase contrast microscope, you can differentiate the different parts of the cell due to the phase difference of the density of the different part of the cells are different. So that makes the observation possible. Phase contrast microscope. And so you need to observe the cells regularly, because for a particular cell, you know that if you are culturing a cell for some time, you know what is its multiplication time. Based upon that, you can see when it is going to be fully confident. But without seeing, you cannot make it certain. Because, for example, you are subculturing a cell just before division. Okay, you are splitting it just before division. What will happen? Immediately after division, the cells will divide and multiply. Now it is double, isn't it? Or in another case, you might be separating cell after the division. So it will divide after 24 or 90 or 36 hours. So it can vary. So you need to observe the cells regularly. And also there could be certain contaminations. Normally we can observe the contamination without the microscope, because when there is a contamination, what happens is the pH of the medium change. You know that there is a color, that's it, you saw that, it's a pink color or reddish pink color, because a pH indicator is added to that. So when the pH changes, the color of the medium also changes. From the reddish pink color, it will change to yellow color. In some cases. But in some certain infections, you will not see that color change. You can see only through the microscope. So regular observation is needed. And you cannot stain the cells, because normal staining process will kill the cells. But there are certain vital dyes. You can just add the dye to the cell culture, and the cells will take in the dye. And the cells, it will not kill the cells. So that type of dye can be used. Then phase contrast microscopy, I have already said. Confocal microscopy, another latest developments in microscopy, but it is very expensive. In a normal lab, we cannot afford it. Electron microscope, that also provide detailed structural features, isn't it? But that also is not available in a normal lab because of its cost, as well as its observation under an electron microscope need detailed processing of the cells. So that will take a longer period of time. In a routine observation, we cannot do that. So routine observation, we will be using phase contrast microscope. Phase contrast microscope, you will see only one plane. You will focus the lens to a particular plane. You can see only 2D pictures. But in the confocal microscope, the different focal plane can be seen at the same time. So you can have 3D images And also if it is a tissue, the metabolic changes at different positions will be different, isn't it? So that sort of observation is possible because it will be looking into the different focal points. The 2D versus 3D versus ID MOOC. So 2D culture, that is a normal cell culture. You are using a culture flask and the cells are growing on the bottom of the flask. So it's a 2D culture. And it has got certain disadvantages. Do not give real tissue situation. In the real tissue, there are multiple layers of cells, isn't it? And also there is possibility that there will be more than one type of cell. So the interaction between these different types of cells is quite important in the case of many of the organs. Isn't it? If you see liver, you can see different types of cells. If you see pancreas, different types of cells, isn't it? Brain, different types of cells are there. So their collaborative working is important. So when they are together, there should be some changes in the physiology as well as the biochemistry. Biochemical changes happening in the cell. So that you cannot study in a 2D cell culture because there is only one type of cell and there is only a single layer of cells. Then cell-to-cell contact inhibition. That is why, or that is how, our body has a particular shape. When the cells are growing together, they inhibit each other. Otherwise, what will happen? In a culture condition, you know that they keep on dividing. Our body cells will not divide like that because of this cell-to-cell contact inhibition. That is not there in the 2D culture. And also, interaction between different types of cells. That is also important. Then, no directed growth. Because in a tissue, the growth is controlled by different factors. In your body, for example, liver tissue or another part, lung, heart, everything is controlled by different hormones. It is produced in some other part of the body. And that hormone will control the growth of the cell. Or this hormone will cause some certain changes in another organ, and that organ changes produce certain messengers that will go to another organ and control it. That sort of control is not there in a 2D culture. 3D culture is slightly near to tissue structure. The cells are in multiple layers, and sometimes we can even provide fluid flowing through in between the cells using capillaries. So it is slightly more closer structure to the tissue culture. And they can be grown with or without support. So like you can have a sort of scaffold and you can grow the cells in between. So that is with support. Or you can allow the cells grow without this support. They will grow one above the other. So microfluidics, because microtubules will be used in between the 3D culture cells so that the fluid can grow in between. So that flowing fluid will provide the nutrients as well as remove the waste materials. So ID MOOC is a new development. So this is a sort of 96 well plate like thing, and each well you are growing different types of cell. And the nutrient medium, the culture medium will be flowing through this all these wells. So whatever waste is produced or whatever hormones is produced, whatever messengers are produced, it is carried to different types of cells. So that will give a sort of tissue-like structure. So 3D culture mimics the tissue-like structure, able to exhibit differentiated cellular function. Co-culture, 2 or more different types of cells is possible so that you can study about the interaction between the different types of cells. Exactly as in a living tissue. Then can stimulate microenvironment, conditions for tissue hypoxia, because certain cases there can be hypoxia. So when there is a blockage in your cardiac vessels, there is hypoxia. Because the area supplied by those cells will not get enough oxygen and nutrients. So if you want to study that situation, you can use 3D culture. The nutrient gradient, etc. And also, using a flowing fluid, you can control the nutrient supply. Better predict in vivo responses to drug treatment because it is very much similar to a tissue condition. Integrated discrete multiple organ culture. So each of these wells will be having different types of cells. One can be liver cells, myocytes, another may be epithelial cells. Okay. So this in vitro cell culture based experimental model then can study intercellular communication because these wells are different types of cells. And the fluid, the medium will be flowing from one place to another, keep flowing. So they can communicate. The messenger can be carried from one well to another well. So intercellular communication can be studied using this structure. So in the conventional system, as I said before, each cell type is studied in isolation. So there is no interaction between different types of cells. But here in IDMO, there is a possibility of interaction between different types of cells. So that is the basic things about cell culture. So you need a culture vessel, you need culture medium, isn't it? And these culture, the nutrient component or medium component varies depending upon the nature of the cells, from where it is obtained or the function of the cells. And the medium, you are providing all the nutrients. And also normally you add a indicator so that you will see any change in the pH of the medium. The pH is quite important because why pH is important? Cell's metabolism is controlled by enzymes. And the particular cells have enzymes having a particular optimum pH. If the pH is low or higher, the enzyme activity will be affected. As a result, the metabolic process will be affected, isn't it? And if you are studying the effect of certain things, and if you are not keeping the pH constant, what will happen? You may get results. The result can be effect of the drug you have used, or it can be the effect of the pH used. Temperature is important because same reason as the pH. What is it? Enzymes optimum temperature. Not only the enzymes will act in an optimum level. And also I have said about the importance of carbon dioxide. You need carbon dioxide. Normally we use 5% carbon dioxide because it is needed for maintaining the pH of the medium. If you are not using carbon dioxide, pH will change. So that is quite important. 5% carbon dioxide. But certain cells need 6%. Certain cells need a slightly higher percentage of carbon dioxide. So depending upon the pH maintained. And the medium contains different types of nutrients. All the nutrients needed for the cell. And also a buffering agent like sodium bicarbonate. Because even though you are providing carbon dioxide, it will maintain only the acidic balance, right? Because carbon dioxide is dissolved in water and produce carbonic acid. So that way it will maintain the acidity. For maintaining the basicity, you need to provide sodium carbonate or something like that. Then also, the medium can be two types, defined medium and undefined medium. The name itself tells you what it is. What could be the defined medium? We know exactly defined components. We know what is added each thing, what level each thing is added in the defined medium. But in the case of undefined medium, the major components are added in a correct proportion. But certain other things you add or you don't know at what level you need it. Because of that, you add fetal bovine serum, FBS. So that serum provides a lot of growth substances as well as many other materials needed for the normal growth of the cells. That is an undefined medium. Normally, cell culture we use an undefined medium. For making defined medium it's very difficult. You have to add tens of different types of materials in microgram level. So it's not easy. We use an undefined medium because we are adding FBS. You don't know the composition of FBS. It varies from each lot, because depending upon the animal from which you are obtaining it, the composition slightly varies. So when we are defining or when we are doing a very specific enzyme which needs micro level control, you will say the batch number of the FBS. Each batch there will be slight variations. That is also important. Because for a particular experiment, from the start till the end, you might be using one batch of FBS. Otherwise, you may get a different result when you move from one batch to another batch. That is why when we buy these things, we buy in large quantity, not a single bottle. We buy depending upon this money available, 10 bottles or 100 bottles, something like that. So that you will get a single batch of these things. That's quite important.